This is my favorite video game from when I was a kid, X-Men for the Sega Genesis. I loved it so much, I bought 2 copies of it. I probably, over the course of my life, have played this game for roughly 500 hours. I can do the entire first level blindfolded. And you guys might be familiar with X-Men, right? X-Men is this story about a group of mutants that use their special mutant powers to go out and fight crime and solve the world's problems. In the 90s, we were kind of obsessed with this idea of mutation, that there would be some sort of change to your DNA, it would give you special attributes, and then you'll be able to use those to do something interesting. But here's the truth. I'm a mutant. You are a mutant. All of us are mutants, because a mutation is just a change to your genetic sequence. And although when your DNA is copied from cell to cell or from parent to child, when that DNA gets copied and passed on, it's copied very accurately, there are always little spelling errors that come in. After all, you have 6 feet of DNA inside each one of your cells. If you imagine copying all those A's, T's, C's, and G's, there's an equivalent number of letters in an entire encyclopedia set. So a couple of typos will pop in periodically. DNA polymerase does a really good job, but nobody's perfect, and no system can be perfectly copied. When errors in replication occur, you get a mutation. And, just like in those comic book stories, mutations do sometimes change your traits. They give you different attributes. So mutation, as I said, a mutation is a change in the nucleotide sequence. There should have been an A in this position, but now there's a T in that position. And you guys now know that because the DNA, the order of the bases, is very, very important, that's going to control the RNA, the mRNA that's transcribed from that DNA. And if the RNA is different, then you're going to get a different protein at the end. The order of the amino acids is going to be different. Now, to be clear, a protein might have thousands of amino acids in it, and maybe only one of them gets altered by this mutation. That could be a kind of trivial change. It's sort of fine if that happens sometimes. In some cases, the protein might actually act better. It might work better at its job than it otherwise would. This is the raw material of evolution. This is where we get the variation for evolution to proceed forward. But most of the time, mutations tend to kind of be a negative. They tend to leave an organism less fit than it otherwise would have been. Now, mutations can happen to entire genes or entire sections of chromosomes. So you can have a gene that actually accidentally gets duplicated. You end up with twice as many of that gene as you otherwise might have had. Or you might have a gene that's just deleted altogether. You might have a section of a chromosome that gets translocated, it gets moved to another chromosome. So all of these are kind of large mutations that occur. But what we're going to talk about are point mutations. Point mutations occur at just one letter in that nucleotide sequence, one letter in that DNA molecule, and even that one letter change can still have very, very profound consequences to it. Okay, this kind of problem should look familiar to you guys by now. We have a template strand of DNA, we have a coding strand for you to fill in, we have an mRNA strand, and we have the polypeptide to translate out. I've added a little extra hitch to it this time. Before, I wasn't asking you to know about the 5' and 3' directions. We were just trying to get the base pairing right. But now I think you're at the point where we can add in this extra nuance of the anti-parallel nature of DNA molecules. So this template strand is running 3' to 5'. Now the other strand should be anti-parallel to it, running backward. So you're going to want to write in which end is which when you solve this problem. Go ahead and take a moment and solve this problem here. Fill in the nucleotides for the coding strand, fill in the nucleotides for the mRNA, and then see what the polypeptide would end up being. Go ahead and fill it in now. Okay, so hopefully your answer looked something like this. If the template strand is running 3' to 5', then the coding strand, the opposite strand of DNA, is running 5' to 3'. That's what anti-parallel means, that they're backwards relative to each other. And that will be true of the mRNA molecule as well. It is also running 5' to 3'. Okay? Now that's important because the 5' end is where we start translating this mRNA molecule from. So this, we're going to start here, and then we're going to work our way down the line. Now, let's get the letters in. Okay, hopefully that looks right. Okay, now that I've filled in both the mRNA and the coding strand, I can use them in order to check each other. 5' to 3', 5' to 3'. AUG, ATG, that looks good. UGU, TGT, that looks good. TTT, UUU, okay, that makes sense. AGG, AGG, that makes sense. Okay, so we got the mRNA sequence here. Then, in order to translate, you'd need to check your genetic code. And you would find that AUG is methionine. It's our start codon. UGU is cysteine. Keep your eye on that one in just a moment. UUU is phenylalanine. That was one of the tRNAs I used in the example. And AGG is arginine. Okay, now I got these dots here. This is just a segment of the molecule. It would continue on after this, but we're only interested in these 4 codons, in these 4 codons here and the 4 amino acids that they code for. Now, imagine this is a section of DNA inside of a skin cell, and someone goes out on a bright, sunny day, they're really excited to go outside, and they don't put any sunscreen on. So ultraviolet radiation comes and shines down on them, and the problem with ultraviolet radiation is it's got high enough energy to actually damage your DNA. So here's something that can happen. The ultraviolet radiation might shine down. So this nucleotide base pair gets knocked out, and it's going to get replaced with something incorrect here. It's going to get replaced with, let's say a GC pair. We're going to replace it with a GC pair. Now sometimes this kind of damage, this is called a substitution mutation right here. It's a kind of point mutation, but it's a substitution mutation because we've substituted one nucleotide for another one here. Sometimes there are enzymes inside our cells that repair this kind of damage. They put things right again, but some mutations do inevitably get through. And now, when we go to transcribe this message, it's not going to be UGU at this codon anymore. Now, it's going to be UGG. And UGG does not translate to cysteine. UGG translates to tryptophan. Tryptophan, that molecule that does not make you tired at Thanksgiving. That is a myth, but tryptophan is what that translates out to. This particular kind of substitution mutation is referred to as a missense mutation. A missense. A missense mutation. A missense mutation is when you change one amino acid codon to a different amino acid codon. But a missense mutation is not the only thing that could go wrong. So here on this slide, you can see that we have that codon UGU, and it should properly code for cysteine in this particular protein that we're putting together. But if we get a substitution mutation and it changes it over to UGC, well, that's okay. That one's actually fine because UGC and UGU both code for cysteine. If you check your chart, your genetic code chart, you should see that both of them will give you the same thing. It's one of those synonyms. When you have one amino acid codon changed to another codon that codes for the exact same thing, we refer to that as a silent mutation. So we've had two different kinds of substitution mutations here. Silent mutations don't change the polypeptide. We changed the sequence here, but we didn't change the finished protein at the end. So we wouldn't even know that that silent mutation had occurred. The missense mutation is the one where you change one amino acid codon to another amino acid codon. And then finally, there's another possibility. What if we took that third letter and instead of an amino acid codon, we changed it to a stop codon? Ooh, that would be bad. That would be real bad. Because that means that as soon as we got to this point in the line, as soon as we got to this point in translation, it would stop. It would make a half of a protein. Whatever amount of protein had been developed up to that point in translation, that would be the end product. It's like allowing cars to go off the assembly line that are only half built. You just got a couple of wheels and an air freshener and you say, yeah, go, take it out to the shop. Best case scenario, that is going to be a useless protein. It's not going to have any function inside the cell. Worst case scenario, it actually ends up being somehow actively damaging to the inside of your cell. So when we have a mutation, a substitution mutation, which alters an amino acid codon to a stop codon, we call it a nonsense mutation. Silent mutations, there's no change to the polypeptide at the end. Missense mutation, there's a single amino acid that's been swapped out for another one. Nonsense mutation, you make about half of a polypeptide and then you just stop because we changed the amino acid codon to a stop codon. Now, how big of a deal is a substitution mutation? Like, you could see how maybe the nonsense mutations would be really bad, but is a missense mutation really that bad? Sometimes, no. Sometimes it's kind of okay. But other times it can actually be quite bad. So in this example, we have a single nucleotide substitution here. We went from CTT in the section of DNA that codes for the protein hemoglobin on one codon, CTT, and we changed it over to CAT. That, of course, will end up causing the mRNA molecule transcribed from that message from that gene to be altered as well. So instead of GAA, you get GUA. And then if you look at the finished product at the end, as a result of this mutation, you go from a glutamic acid over to a valine. Now, as it turns out, that mutation occurs at a juncture of the protein which is very, very important to folding, a critical fold location. So this creates a misfolded protein, and it's a hemoglobin protein, so it's pretty darn important. Here's a picture of red blood cells behaving with that mutation intact, so you can see that periodically they take on this kind of crescent shape. Now, the normal biconcave shape of red blood cells, kind of like a donut inside of a sock where you have a little divot on either side, that's really important to their ability to squeeze through capillaries and to carry oxygen around. It increases their surface area. So taking on this new shape causes them to clot up, to clog capillaries, to not progress through. This causes a lack of oxygen reaching certain areas of your body. It causes a lot of organ failure. So yes, actually a single missense mutation can lead to pretty disastrous consequences. So those are three kinds of substitution mutations: the nonsense mutations, the missense mutations, and the silent mutations. But there are other kinds of point mutations as well. There's something called a nucleotide deletion or a nucleotide insertion. In a nucleotide deletion, what happens, take a look at this slide here, is a single nucleotide is removed from the DNA molecule. Now, that has a really interesting effect because the ribosomes don't know where one codon was to begin and another codon was to end. They just read 3 letters at a time. So if you remove one of those nucleotides, every letter following that removal gets shifted up in the reading frame, right? So a nucleotide deletion actually changes every single codon that is downstream of that mutation. It would be like if I were to remove a single letter from a book and then shift all of the words, all of the letters forward, but I left all of like the spaces and the punctuation and the page breaks and the titles and the chapter headings all in the same place. So as you kept reading the book, it became less and less comprehensible. Like the beginning of the next word is at the end of the word you're currently reading. It would look a mess. So deletions can lead to what we call a frame shift. A frame shift is when everything moves down one step. Same with nucleotide insertions. And nucleotide insertion is just the opposite. We shimmy an extra nucleotide in where it's not supposed to be, and as a result we have another kind of frame shift where everything moves a little bit further away here. So in both situations, you're going to have a whole chain of incorrect amino acids being produced. So these mutations can be quite bad. And this is why it's really important to avoid chemicals that might cause mutations. Chemicals which we call mutagens. Mutagens are anything that's going to be able to damage your DNA, and when your DNA tries to repair itself, sometimes these mutations manage to get in. A lot of mutagens are also carcinogens because your body has genes which are associated with suppressing the formation of tumors. So if you damage those tumor suppressing genes, you can end up gaining cancer as a result. So a lot of times these mutagens are carcinogens. So where do you find mutagens? Cigarettes have a lot of mutagens inside them. Ultraviolet radiation, you know how it increases your risk of skin cancer? That is a mutagen. Radioactive waste, as of course the Teenage Mutant Ninja Turtles discovered, is also a mutagen. Now mutations are typically quite bad, as I just mentioned. However, every now and then, very, very rarely, a mutation occurs which actually causes the protein that has been altered to be better. It's better at its job. It's got some new ability. Maybe you can digest some new type of food that you weren't able to before. So you have an enzyme that's adapted to a new kind of food source. Or maybe your muscles end up being a little bit more springy than they used to be because of a change in the proteins that create the contraction or that hold the tissues together, right? All kinds of little tweaks, little tiny tweaks, can end up having an effect in the adult organism. Now, obviously, the Teenage Mutant Ninja Turtles, fictional creatures. There, of course, would never be any evidence of real mutant turtles showing up with extraordinary abilities. That would be silly. Before we move on with mutation, I want to show you guys a little project that I worked on a few semesters ago. It's a recreational spreadsheet. You know, everyone makes these periodically, right? You want to have some fun. You say, I'm going to open up Microsoft Excel and make myself a spreadsheet in order to relax. Well, this spreadsheet is a little bit special. If you see here, zoom in, I have an RNA sequence running up here at the top, and it continues on over in this direction. And under the RNA sequence, it says what amino acid would be represented by each codon. And I've programmed the whole genetic code into this. So if I were to change this from AUU over to AAU, then it would change to a different amino acid as well. We'll put it back right here. Now, here's the trick. What I told this spreadsheet was, take whatever is written on this top line here, this first line of RNA, and copy it down here. But there will be a 1 in 100 chance that any given one of these nucleotides in this chain changes to a different letter. Okay, so there's a 1 in 100 chance for each one of these, and you can see that this one here actually changed in that copying process. And because it changed, we got a different amino acid, right? We went from THR over to proline right here, from threonine over to proline. And I had it highlight any kind of changes going down the line. Now, it's relatively rare for a mutation to occur. 1 in 100 chance is pretty low. So let's see here. Were there any other? There were no other mutations in that first copying. But that mutation that did occur, well, there's no way for it to change back once it mutates, so in the next generation, it was conserved. We have that same change, right? It continues on down the line. Each generation, there's another chance for mutations to come out. So here, in generation 6, we get a second mutation where this valine is switched over to an alanine as a result. And we still have the original mutation as well. In generation 7, look at this, we get 2 new mutations popping in with just a 1% chance likelihood of each one. We got 2 new mutations, so we are up to 4 here. Every single generation is going to have some mutations that the previous generation did not have, and they are going to be copied from generation to generation. So not only are mutations, even though they're relatively unlikely on their own, over the long course of human history, they are inevitable, and they will continue to accrue and acquire and be carried forward through time. Let's see here. Are there any good? So we've seen quite a few missense mutations pop up, one amino acid being switched to another one, but here we got a nonsense mutation coming in. We went from AAA to UAA, which changed it from lysine over to a stop codon. And I should say that this chart here is random. So you can see where it highlights all the... Ooh, that's too far out, isn't it? Is random. So you can see where it highlights all of the mutations. Every time I hit the delete key, we get a new set of mutations coming through. Sometimes they show up early, sometimes they show up a little bit later, but inevitably, down at the bottom, you're going to see a lot of mutations showing up. So we got 2 here in this final set, but let's go. Let's randomize it a bit. Look at that. Every single time, just based on a 1% mutation rate, we get tons of mutations showing up after 25 generations. So what is the point of all of that? Mutations are inevitable. They are going to happen. DNA is going to copy itself as is. So once those mutations happen, they will be passed on to new generations. And those mutations will cause changes to the proteins that are created in those organisms, and changing those proteins will change the organism's traits. So organisms will inevitably change over time because of this mutation process. Once they change, there's really no way for the DNA to change back again. Yeah, it is possible that we could have a mutation that changes one way and then it mutates back and changes again. But it's infinitely more likely that you're just going to get more mutations instead. So we're going to move into the evolution section and keep this idea in mind, that not only is mutation likely to happen, it's going to happen, and it will have an effect on each generation. Each generation will be different than the previous generation.